Hi everyone, and thanks for coming. Welcome to this month's Machine Learning and Science seminars, where today we're going to be talking about machine learning modelling in life sciences. Today we've got two speakers from the School of Maths and Stats, Professor Colin Torney and Professor Dirk Bussmeyer. Colin will be talking to us today about machine learning methods for modelling animal groups on the move, and Dirk will be talking about machine learning for modelling complex cardiac physiological systems. So without further ado, I'd like to invite up to the stage our first speaker, Professor Colin Torney.
Great, thanks very much. Thanks for coming. So today I'm going to talk about how we're using machine learning to study animal populations. And mainly I work on these animals here, which are wildebeest, which I'm sure you've all seen in nature documentaries. These are the ones that cross the rivers and get eaten by pretty much everything eats them. But so I'm going to talk about two separate topics. The first is a bit more applied, actually, getting out and looking at these animals and attaching devices. And the second is a bit more theoretical, how we use machine learning and neural networks to analyze movement data. We'll see how we do for time. So I'll go through, and if we don't get to all of it, we can just skip through. So mostly I work in Tanzania, so the applied aspect of my research is in Tanzania, and I largely work in the Serengeti National Park, which is this region here, and this is home to the wildebeest migration. So this is approximately 1.3 million animals that make this circuit each year, going from the north down to the south and back up again, just following the rains and moving around to get the nutrients they need to survive. And they also play a really important role in the ecosystem as a whole. They're the drivers of the whole system. Even so, without the wildebeest, you wouldn't have the lions, you wouldn't have the predators, you wouldn't even have a lot of the lower, the insects and the smaller animals, because they really just keep this ecosystem functioning. So it's obviously important ecologically, but also very important for the region in terms of the economics. It's the main driver of the economy in Tanzania, or one of the main drivers is tourism. So we've been working with Tanzania National Parks, and they're the governmental organization that manages national parks across the country, and also the Frankfurt Zoological Society. So they're a conservation organization that have a long history working in Serengeti and Tanzania and across Africa. And these two organizations have been developing a LoRa gateway array. So this is a long-range radio network. So it's very similar to mobile phone towers, but it's lower power and longer range. So you don't need as many towers, and your devices can be relatively efficient to run in terms of their battery life. They're primarily using these gateways to monitor rhinos and vehicle traffic. So they want to know where all their vehicles are. So they've had this array of gateways. You can see this cluster here. I mean, mainly they're putting these gateways where the rhinos are. So they want to monitor these rhinos and get this live feed of where the rhinos are, so they can, if there's any incidents, they can be there quickly. But we've been working with them to see, can we leverage this infrastructure to use it to study the wildebeest population, the wildebeest migration. So this is how they're currently using it. They have this live feed of vehicles. They know where the rhinos are, so they know if there's any issues, they know which vehicles are in the area, and they can just radio and move rangers around. So we're trying to use this system for studying the migration, and we do that by attaching devices, collars to the wildebeest, which tell us, historically they tell us the location. So they have a GPS, which is here. This is a little GPS device. And the current devices give a fix 2 or 3 times a day of where the animal is. Now we're looking to increase the amount of information we can learn about these animals as they migrate. So we're trying to go beyond just location and introduce behavior through an accelerometer and a gyroscope, and also increase the amount of fixes. So we don't just want to know where they are 2 or 3 times a day. We'd like to get hourly fixes or even more. So initially we sort of tried to work with different companies to see if they could manufacture something that would work for us. We couldn't really make any headway with that. Companies have their specific devices. They're not so interested in customizing them. So we ended up developing our own system that had the functionality that we needed. So it has a solar panel. We have a little solar charge controller which controls the battery charge. This is the microprocessor unit, which is an Arduino unit. And we have this accelerometer. So we put all these components together, and this gives us everything we need to study these animals and get the information we want. And the really nice thing about these Arduino components is that you can actually put machine learning algorithms and run them on the chip, on the device. So you can shrink these machine learning algorithms down and run them. This is edge machine learning, where you're processing the data on board. So we put together this very small convolutional neural network to process the accelerometer data. So we're getting a feed from the accelerometer of the acceleration of 3 axes and the pitch and roll of the head or of the collar. And if you're familiar with convolutional neural networks, I think this will be quite a trivial example for you. I mean, it's just 3 convolutional layers and max pooling in between, and we're really constrained by how much you can fit onto this very small microprocessor. So you can't have a very large neural network. But this we found worked reasonably well. So we train the model normally, with a GPU. When you have your weights, you have your architecture, then you just load that onto your microprocessor, and then that runs and can predict on board. So we need training data to train this model. So Serengeti wildebeest are not so easy to follow. They move large distances, and there's really large herds. So once you put the collar on, typically, initially they're very excitable, very panicked by the whole process. So they run, they jump around, they go crazy, and then they just disappear into the herd. So even if you could follow them, you could get them for maybe a few hours, but once they're gone for the day, it's really hard to relocate them. So instead of working in the Serengeti, we went to a different population. This is at the Kilimanjaro Golf and Wildlife Estate. So this is the Serengeti is over here. So it's close, but it's more of a sort of a tourist, well, more of a sort of it's basically like a golf estate that has some wildlife for the golfers to go and look at when they're having a day off on the golf. Very nice place. Very wealthy place in terms of some aspects, but also in terms of the animals. They don't have a huge amount of money to spend looking after the wildlife. So you can see the contrast. These are the golf courses, amazing, green, lush, well watered, and this is the region where the wildebeest are. There's been quite a severe drought the last few years, but the wildebeest really they're not getting much to eat, and they're in this very the population is too dense for the region, effectively, so they are struggling a bit. But we went there before Christmas, not last year, but the year before, and we put out these collars on these animals, and then we could go, because we knew they're in quite a small area, so we could drive to that area, drive around, within 5-10 minutes we could find the wildebeest and watch them. So yeah, so this is the wildebeest with a collar. We put the collars on so we know which collar we're looking at, the ID of the collar we're looking at. And yeah, we had 6 collars in total. We got data from 5. One of them we never got data from. One of them died fairly quickly. So some of the animals spent a lot of time in these wooded areas and just didn't get sunlight. And without the solar charge, then the batteries just died. But the others worked for around 2 weeks. And then we spent 2 weeks just following the animals. We had we recorded them with a video recorder, and we also had sort of live monitoring of what they were doing. And then this is the live stream of the accelerometer data. So there's like 5 readings, which is like the X, Y, Z acceleration, and then the pitch and the roll. This is the orientation of the collar. And then we had this live stream coming over Wi-Fi network. So if we were close enough, we could get the raw data, and we could use that for training. So we spent a lot of time just following these wildebeest around in this area. So we'll see wildebeest soon. Yeah. So we're just watching them as they're walking, we're following the one with the collar. We pick which one we're going to do for that day or the morning or afternoon. And then someone's close to it trying to get the signal, trying to get the data downloaded, and someone's recording it, and someone else is like monitoring, lying, walking. Skip one's quite loud. So we have all that data. We ended up, I can't remember the exact numbers, but it was around 50 hours of footage and sort of time series data and labels. And then we trained our neural network on these data. This is the results from the validation data where so we have 4 classes we're interested in: grazing, lying, standing, and walking. So the main idea around the project is trying to understand the energy intake of the animal, how it's getting on during the day, and what the disturbances within its ecosystem are doing to its behavior in terms of its probability of survival. So if they're in regions where there's a lot of disturbance, maybe they're not spending much time grazing. So then from that, if we can get a sense of how much time they're spending grazing or lying in certain areas, we can figure out, okay, in these regions, they're really not getting the energy they need to survive, so they have to go to these regions. These are the safe havens, and then try and accumulate that over time and make predictions about how much disturbance this population can tolerate before you start to see significant declines in the abundance. So the main thing to note would be standing and walking is there's a bit of confusion around that, but we think that's really because we use a 10-second window and they're constantly alternating. If you look at the time series, it's really clear when they're walking, when they're standing. So that's probably not to do with the algorithm. It's more to do with how we've set it up in terms of every 10 seconds has to have a single behavior, even though there might be 2 behaviors, in fact, in that window. And then the standing and the lying, we were a little bit surprised that we could tell the difference. But that seems to be because of the head position. So when they're lying, the head is raised, and when they're standing, the head tends to be more down. And the main issue we're a bit worried about that is because in the Gulf estate, there's no predators, so they're not vigilant that much. And when they're vigilant, the head tends to be up. So I think when we go to the real ecosystem, we're a bit concerned that there's the standing, lying is, there's going to be a confusion between lying, standing, and vigilant that we don't capture in this population because they're not vigilant that often, because they have no predators to be worried about. So we took this trained model, loaded it on collars, and then we deployed it on the Serengeti population of wildebeest. This is in April of last year, so roughly a year ago now. So these collars were collecting the data on the location and the behavior. So they're solar powered, and they transmit data to these gateways, these LoRa gateway arrays. So we get a fix every hour, and then we don't classify behavior all the time. We just do 30 minutes every hour, and that's just so that we have time to transmit the data at the end of the 30-minute period. We have 30 minutes of classification, then we give the collar 20 minutes to transmit the data and anything it's got stored. And then every 10-second interval, we're classifying as one behavior, one of these 4 behaviors, and then we encode it as binary digits. And this, the reason I'm saying is this, we really have to shrink this data down to be able to transmit to the LoRa array. And so we get it down to like 30 minutes is encoded in 45 bytes of data, and that's within our. So we have these regulations about the use of LoRa in terms of how much data you can send at different data rates. So the rate tells you how far it can go, or controls how far it can go. So if we want to get the really long range communication, then we have to send a small amount of data. And there's a trade-off between the range and the amount of data you can send. And the main reason I'm highlighting this is that because the edge machine learning really allows us to take this high frequency stream of accelerometer data and then condense it down to something we can transmit over LoRa, which we wouldn't be able to do just with this raw machine, with the raw accelerometer. So this is us out in April last year, putting the collars on. As I say, we deployed 8 collars, and once once we deploy them, the wildebeest are basically just off. We're not going to see them again. Some of the other collars we have VHF, because we can actually track them and find them, but these ones we don't. So we only know where they are based on the law of communication. And yeah, this is just the solar power. This is the another angle. You can see the solar panel on top. So we're getting this data now. It's coming through. So the first thing we looked at was, can we just plot something that just shows us this is making some kind of sense? It's not giving us complete nonsense. So what we looked at was the distance. We have the hourly fixes. We looked at the distance between these fixes and what portion of time the classifier was telling us the animal is walking. So if it's telling us it's walking 100% of the time for 30 minutes, we don't want to see, okay, well, actually, it didn't move at all in that window, because that doesn't make sense. And similarly, if it's saying that it didn't spend any time walking, we don't want to see long distances. So we plotted this and said, okay, yeah, this is plausible. This makes sense. We're only seeing half the time. So, we know what they're doing for 30 minutes. They could spend the other amount of time, really, they could spend 30 minutes lying down and 30 minutes running. So it's not perfect. And also they could be moving back and forth, which you wouldn't pick up on the GPS. But overall, yeah, this is something we would expect. That's good. And then we also looked at the other activities over the course of a day. And we were quite pleased to see that it does, it's quite noisy, but we do see patterns that we would expect, and that makes sense biologically. So just looking at the blue line here, this is the amount of time spent grazing, or the fraction of predictions are spent grazing. And what we see is two clear peaks. And this is just after dawn and just before dusk, or at dusk. So this is what we know, how we know the wildebeest actually behave. They have these grazing bouts just at sunrise, and during the day when it gets really hot, they tend to be more static, and they spend a lot of time lying down. So we see these peaks as we would expect, and we also see sort of lying down at night and more this daily pattern, which lines up with what we, from the ecology, from the biology, how we think the animals would behave. So our main issue now is we have a bit of a problem in terms of the robustness of the collars. We're trying to figure out. So what I'm showing you here, this is the last known receipt of communication. I downloaded this before Christmas, but so the latest date I think is November. But you can see that some of these collars, we heard from in April, but we never heard from again. And some, you see those October, August, May, but we're seeing this quite rapid drop off in terms of how long these collars are lasting. And we don't actually know, because a lot of these gateways, they're quite sparse. We don't know if animals are just out of range for a long period of time, or the collars just failed. So we're kind of constantly watching these data come in and see if they come back in range, or has the collar just stopped working. So that's the main focus now. We have this proof of principle that we know we can do it. It does work, but the caveat is that we're not seeing the robustness we need in these collars. So we're going to sort of the next stage, which is redesigning the board. We're a bit concerned that the battery we're using the quality isn't high enough. So we've redesigned the board, and we're trying to, instead of using these individual components, we're trying to just embed everything on a single PCB and see if we can get a more robust collar that's going to last. Because it's only worth doing if it's going to last 2 years. Otherwise, it's not worth the time and effort to collar just for a few months worth of data. So I did have a second section. I think I'm just going to stop there and take questions, because we're pretty much close to time.
A few minutes if you want.
Shall I just briefly go through it? Yeah, so this is more of a theoretical topic. So a lot of the work we've been doing is once you have these data, is how you try and analyze and how you understand it. And one of the methods we've been keen on, and has been pleased with, that's given us some success, is graph neural networks. And this is just a generalization of neural networks to data that has this graph structure or network structure. And it's very similar to the transformer architecture. So, I mean, I'm sure a lot of you will have been familiar with transformers and graph neural networks, but for anyone who isn't, it's, the graph neural network just allows you to think about structure in your data as a network, and the transformer effectively is like a fully connected network with attention. So this is the attention mechanism. And so why these are working well for us is that there's a lot of similarity between these types of models and the models we use to simulate collective movement. So when we simulate collective movement, we have an individual that pays attention to its neighbors and it updates its state. So it sees the position and velocity of its neighbors, and so that's sort of the V in the equation previously. That's the values of the individual, and then it updates them according to some attention-based rule. Okay, and so this is a very simple model, the VSEC model, where you just look at your neighbors within a zone of interaction, fixed radius, and align with them. And there's also more complex models where you have repulsion, attraction, alignment. And you can simulate these models and you get really nice, visually appealing simulations that look like real collective behavior, but they're not really grounded in the data. But we're using these models to see, can we take the machine learning approach, deep learning approach, combine the models with data, and get simulation models that are more grounded in reality, more empirically based. Let's skip this section and just go to the simulations. Yeah, so one of the nicest things about these graph neural networks is that you can get very realistic simulations of physical systems. Let's click the play on that. So this is not my work. It's a paper that was out a couple of years ago now, 2020, looking at how you can, instead of simulating a set of equations, a set of partial differential equations, you can just train a graph neural network on data, run that forward, have the graph neural network make predictions of the next step, and then keep repeating that process, and you get something that looks very similar to the real physical system. Next one. You might just have to click outside the. Yeah, that's it. Yeah, so I mean, the concept of the transformer, these attention mechanisms, is that the attention is based on the value of the neighbor or the individual, the node. The node has values, and you pay attention according to how it's behaving. In these models, the edge is really important. So it's not so much about what the individual is doing; it's where it is relative to you. So you might follow an individual that's close to you, but ignore an individual that's far away. So we really need to build in this idea that the edges matter in terms of the model we're using. And so there's been recent work in this area, so adding this edge property into the architecture. So we've been using this to simulate these classic models of collective movement. So this is on one side, it's the simulation, and then you give the simulation results to the transformer, and then ask it to run its own version of the simulation. And so on the left is the real simulation, on the right is the GNN. So it's able to pretty much reproduce it, and then as you change the density, you get different behaviors. So there's this transition from disorder to order, and the neural network can really learn about that and match that behavior. And the nice thing is that if you train the model on one density, so here the model is trained on just the middle density, it can extrapolate. So it can, without being told there's a phase transition, you train it on one density, and then if you give it different densities, it makes accurate predictions in terms of how the system would behave. This is how the system would behave if the density was higher, and it gets it correct even though it's never seen that. And yeah, so the next stage of this is actually really just applying it to real data and dealing with the challenges of real data where we don't have periodic boundary conditions. We have physical barriers and all these issues that we're just trying to work on now. Okay, and that's really the end.
Thank you, Colin, for the great talk. I think we have time for a few questions, if anyone has any. Okay, so I have a quick question just about the sort of extension of this work and if it could possibly be extended to track perhaps critically endangered animals like rhinoceros you mentioned earlier.
Yeah, I mean, we are actually working with TNAPA on the rhino project to see if we can use the same collar on the rhino. And to be honest, whenever we talk to people in Tanzania about these collars, they always say, well, can it work on rhino? And that's always the first question. No one's so interested in the wildebeest for some reason. I don't know why. But the rhino, yeah. Yeah, I mean, the tricky part of the rhino is it has to be on the ankle. And also the training data. We're not sure how we're going to get the training data because it's a lot harder to tag a rhino, and you do it once and they're a bit more fragile than the wildebeest and highly valuable. So just a bit wary of we don't want to have to put a collar on, take a collar off, and then put the trained collar back on. So that's a challenge now.
Thank you. Any questions? Yes.
Thank you very much for the talk. I was wondering, in the case, you mentioned, for example, that sometimes you don't have predation and that has an impact on the generalizability of this approach. I was wondering more broadly, I suppose it's more sort of biological, ethological question. To what extent do we think that these collars are going to affect the behaviour of the wildebeest and how, or, got footage where you're watching them sheep, I appreciate that's not generally the case. Do we know if there are any behavioural changes associated with the collars that we
Yeah, I mean, so there has been quite a bit of work on this. I think there's no behavioural changes. I think the main issue people have seen is that it does increase predation risk depending on the colour of the collar. So one of the main significant findings is that if you put on very brightly coloured collars, so the collars that people typically use, I mean, mostly in the US for deer monitoring, they tend to be very brightly coloured. So people have in the past taken those to Tanzania and put them on wildebeest, and the predation is just instant. So I think the study, as far as I know, I mean, these are very lightweight collars, and the modern ones compared to what was being used 20 years ago, it's a huge difference. So we think they do get used to them fairly quickly, but predation risk, if the collar is conspicuous, is an issue.
Hi, thanks a lot. So I've just got a question about the generalization to different densities for the graph neural network. Do you know to what extent it can generalize? Because there's some work that I've done with graph neural networks for other sort of similar-ish problems. They can generalize to, let's say, an order of magnitude, more nodes than what they were trained on, for example. But above that, they sort of lose the ability to generalize. Have you found anything like that, or is that?
Yeah, no. I mean, so I guess in this example, it's a little bit easier because once you get past this transition, then it's just aligned. So if you look at this order parameter, the difference between 1,000 individuals, if they're aligned, and 1 million, it's the same. So but yeah, it just sort of asymptotes. But there could be sort of, I think if you really went into that tail, you could see that maybe it wasn't quite picking up these changes at this large level.
It's also the case. I guess it's also the case that they're not sort of expensive enough computationally to train. You have to worry about needing to train on really small numbers of nodes.
Yeah, no, I mean, I think the main reason for this wouldn't be about computational efficiency. It'd be more that you could learn about the behavior of a complex system in one environment and then translate it. So you think like human crowds, okay, we have this human crowd. What happens if we put a pillar here? How does that change the dynamics of the crowd flow? So it would just be sort of, can it capture those types of behaviors rather than saying, okay, it's really expensive to simulate or to train on these densities. But, yeah.
Thanks again, Colin, for the great talk. So now, time for our second speaker, so I'd like to welcome to the stage Professor Dirk Vismaier.
Okay, thank you very much. We're making our big jump from birds to hearts, talking about the application of machine learning in cardiac physiology. So in the last 10 years or so, there have been impressive advancements in the mathematical modeling of complex systems, in particular sort of cardiophysiological systems. Here are sort of 3 more recent book titles. And that helps us to get better insight into the physiology, the pathophysiology, and potentially suggest better treatment options. But it's fair to say that they are not routinely used in the clinic yet. And basically we'll be talking about the challenges. I mean, the first challenge, obviously, if you have a very complex model, then you need to be able to solve it. These equations don't have closed form solutions, but we can use powerful numerical techniques like finite element discretization to numerically solve these equations. That's the so-called forward problem. But once you've been able to simulate from the model, then the next question is, well, how can we now calibrate the model in light of particular data you get from a patient in order to make patient-specific decisions? And that is the so-called inverse problem. We want to go back from the data to the model to infer the parameters. And that is where machine learning and statistics comes in, and that is basically what I want to talk about. So that is a roadmap of what I want to cover. If we manage to get through that in 25 minutes, I don't know. Let me start with the motivation. I want to talk about sort of machine learning methods, basically take you on the journey that I've been on with my group, starting from more basic machine learning method, then to more complex one, and also talking about graph neural networks that Colin has already started talking about. The motivation, well, it's a simple one. According to the BHF, the British Heart Foundation, a quarter of all deaths in the UK are related to cardiovascular diseases. We have on average 460 deaths each day. And here is a cross-section through a heart of a patient who's died from a heart attack. You see the scar tissue at the bottom. That is sort of tissue has actually died as a consequence of deprivation of oxygen. And then the physical properties of the tissue change. The physical properties in terms of its flexibility, its contractility. Now, a large proportion of patients who get a heart attack don't actually die. The treatment would gain enormously by having better insight into what actually has changed in the tissue as a consequence of the infarct, what has changed in the scar tissue. That's basically what we want to try to understand in order to then advise and come up with better treatment options, have a better insight into the sort of damage that has been done to the cardiovascular muscle as a consequence of the infarct. Well, we can't extract the heart, we can't study the heart muscle itself. What we can do is run magnetic resonance imaging studies. Here's a cross-section sort of a cardiomagnetic image. That is sort of a static image, but obviously, well, the heart is in operation. It's contracting, expanding, and contracting, expanding. You can see the movement and the contraction of the heart in that image. I have a video, but it's not embedded. So I think in the interest of time, let's just leave it with it, so don't switch. But you know that the heart isn't static. It's moving. And the important thing is that the movement is controlled by forces. We can't measure the forces, but we can develop a mathematical physical model that relates the forces to the movement that we can see. That interaction depends on the tissue parameters, the cardiovascular and the constitutive parameters. And in principle, we can get to the parameters by studying the motion. So that's the whole idea. So we can't measure the forces, we can't measure the parameters directly, but if we see the motion, then we can infer the parameters based on the motion. That is the basic idea. But in order to do that, we need a particular model of the cardiovascular structure, and that is what I want to basically talk about next. So we start with the segmentation of the left ventricle. So on the left, you basically see the magnetic resonance image scan. Then we want to extract the left ventricle. That is the larger of your two heart chambers that pumps blood enriched in oxygen into the body, which is particularly vulnerable to cardiac infarct and myocardial infarction, heart attacks. And you see sort of a structured model of that heart with a mesh, a discretized set of nodes that we need for the numerical solution of the mechanical equations. At the heart of it is a partial differential equation model of the cardiomechanic processes. I don't have the time to go into that. That was developed by my collaborators in our applied mathematics group. It's basically a set of partial differential equations describing the cardiomechanics. But if they can solve these equations, then you can take the heart at an early time point t1, that is early diastole, and then you can actually follow the movement and predict the state of the heart, the left ventricle, at a later time point t2. And we're looking at diastole, that is the passive filling process. So when blood is pumped into the heart expands. At the end of diastole, now you have an electric pulse, and you have contraction causing now the next phase, systole. We are not modeling that in our group. We're focusing just on the mechanics. So the first half of this pump cycle. The expansion, the state at a later time point, depends on the material parameters. And then we solve the equations with finite element discretization. And then if you know the material parameters, then you can communicate that to the clinicians. Then they basically can get deeper insight into potential damage done to scar tissue. But the problem is we don't know these parameters. So how can we get the parameters? Now, in the lab, you could measure that by pumping blood into the heart, and then you are looking at how the heart muscle expands, how the left ventricle expands. And you can measure the stress as a function of stretch. That is sort of similar to a simple spring, which you know from, your highest physics. I mean, it's a simple law between that stretch, that is the expansion of the compression of the spring, and then the force, that is stress. And in sort of a simple regime, it's just a linear relationship. Now the heart is more complex. It's a three-dimensional object, so you have different directions. And also, the law is not just linear. For elastic material, it is linear, but typically for large expansions, then you have deviations from the linearity. The heart is actually a viscoelastic material, so we want to model the non-linearity as well. Now, in principle, if you now know this stretch-stress behavior, then you can get the parameters. They're really sort of related to stress and contractility and flexibility parameters along the different directions of the heart muscle. And that tells you a lot about the physiological composition of the material, potential damage done to that. Just that we can't do that in vivo, we can only do that in post-mortem studies. So what do we do in vivo? Well, we need to resort to statistical inference. So again, at the heart of our model is the cardiomechanic model. We take as an input the shape of the left ventricle at an early time point, early diastole, T1. The model depends on the constitutive parameters, which characterize the tissue properties, and then the pressure of the blood pumped into the left ventricle. Then you use finite element discretization, and then you can predict the state of the left ventricle at a later time point. We get the left ventricle at early diastole from the magnetic resonance images. So either by segmentation manually, which is still state of the art, or by automation using the deep neural networks or convolutional neural networks. That's what we are also working on, but that's not the topic of this talk. We want to get the parameters, and we get the parameters by quantifying the mismatch between now the shape of the left ventricle at late diastole at time point t2, comparing that to the prediction from the model. We quantify the mismatch and find the parameters that minimize the mismatch. Well, the question is now how to quantify the mismatch. When I started working in this field, the state-of-the-art, the textbook method was that you looked at various landmark points, indicated here in yellow, and then you look at how these landmark points move as a consequence of the expansion of the left ventricle. That displacement is what's called a strain. So my collaborators looked at 24 strain positions. There are different ways how these landmarks can move, and my collaborators focus on the circumferential strains because they are particularly easy to extract from the magnetic resonance images. So what we have are 24 circumferential strains that are extracted from the magnetic resonance image, and we can also easily get the volume of the left ventricle at late diastole. So that gives us a 25-dimensional parameter vector. We get the values from the magnetic resonance images. So again, 24 circumferential strains and the volume of the left ventricle. We can predict that with our model. The model depends on the shape of the left ventricle. It also depends on the cardiomechanic parameters. And we want to now infer the parameters by minimizing the mismatch. That would allow you to minimize the objective function and find the best parameters. It doesn't tell you anything about the uncertainty. In order to do uncertainty quantification, we want to go one step further. We want to get the whole likelihood. Then we can run Markov-Chain Monte Carlo simulations. In principle, that requires you to use a noise model. Here you see a simple IID Gaussian noise model, but it's easy and so straightforward to expand that to allow for correlations between the different circumferential strain positions as well. So in principle, you get these parameters now, but there's a catch. The catch is the fact that you have to run forward simulation from the model whenever you change the parameters. The typical simulation takes about 15 minutes. So to get the parameters, optimize the parameters, takes about a week in computation time. So if you then want to run a full-blown MCMC simulation, that goes into the order of a month. So that is not feasible for the clinic. So that now basically is where my talk really starts about machine learning methods. And these are sort of all different methods where we build different emulators to help with the computational complexity. I start with the simplest one that we started working with, Gaussian process emulators. So our mathematical model is an abstract representation of the real world, but it's computationally too expensive. We want to have a second level approximation, approximating the mathematical model with a statistical model or machine learning model. It's called an emulator. We started using Gaussian process emulators. Here's sort of a seminal paper by Mark Kennedy and Tony O'Hagan, written over 20 years ago. So the idea is a simple one. You take your parameter space, you cover it with a grid in the simplest case of a two-dimensional space shown here. Then you look at the model outputs. Remember, we've got 24 circumferential strains and the volume of the left ventricle at end of diastole. You run the forward simulation in advance. If you have a higher dimensional space, you won't just take a simple grid structure. Use a space filling design to uniformly cover the parameter space, typically taking a Latin hypercube design or Sobol sequence. And you run your forward simulations. You have now all the outputs. You want to get from a set of discrete observations to a function that is an infinite dimensional object. We use the interpolation to get now from the individual observations, individual outputs to the whole function. And so we used Gaussian processes. Gaussian processes have become popular for the following reasons. First, they're flexible, they are nonlinear models. You specify the prior knowledge where you have it in function space and not in parameter space. They allow you to easily quantify the uncertainty, but one of the main reasons is what we are doing here is we have a deterministic model. Right? The cardiomechanic model is a deterministic model. The Gaussian process does exact interpolation, not just regression, like a neural network, but exact interpolation. That's why they have become so popular in this field of emulating deterministic systems. Okay, that is work done by Vinny Davis and Roberto Noe. Vinny was still a postdoc in our group. Now he's a lecturer in our school. And here you see sort of the stress behavior comparing now the literature value. Well, the literature was actually the simulation study done by my collaborators based on inferring the parameters with the full mathematical model after 1 week of computation time. And then the blue line, you don't really see a difference because they're effectively completely overlapping, shows the results with the emulator. And so the upshot is that they get a very accurate model and substantial reduction of the computation time from 1 week to 50 minutes. So that was an encouraging start, but there's a fly in the ointment here in that, well, this is oversimplified, and that we only focus on the constitutive parameters. We have completely ignored the shape of the left ventricle. Every patient, every person, everybody in the audience has a different shape of the left ventricle, of the geometry of the heart, and we've completely swept it under the carpet. So we need to integrate the left ventricle into the emulator. And you see that is a challenging task because you only look at 4 constitutive parameters, and building an emulator in 4-dimensional space isn't so challenging. But then our left ventricle is described by a 17,000 dimensional grid, the high-dimensional vector that has to be integrated into the emulator. So that leads now to the next task of dimension reduction. And we started with the simplest approach, PCA. So you look at the population of, we had 150 volunteers and MI patients, myocardial infarction patients, so patients who had a heart attack. And then we take magnetic resonance image scans for each patient. And we extract the left ventricles from the magnetic resonance images. These are these high-dimensional, 17,000 dimensional vectors. We get the covariance matrix, get the leading eigenvectors, and then do PCA. So we are projecting now our representation of the left ventricle to a low-dimensional representation. We can build an emulator in this low-dimensional space, and we go back from this low-dimensional representation back to the original representation. U is the vector, the matrix composed of the leading eigenvectors, and then we do the inverse transform. That's what it looks like. What you see here is the representation of the PCA component after bottlenecking it, right? We take the left ventricle, bottleneck it through this low-dimensional representation, go then back to the original space. Then you see the gray and the dark gray shaded area. That's the representation of the left ventricle with its outer wall and its inner wall. And then there's the red grid structure that is just the original, taken from the original image, and the gray shading is what we get with the principal component reduction. You see that overall the agreement looks okay. We have various deviations here. And overall that agreement looks okay. But obviously the question is, what is now the knock-on effect on inference when we want to do the inverse problem and get the parameters? Number of principal components. Well, if you use more components, you have a reduction in the reconstruction error of the left ventricle, but an increase in the emulation error. So that needs to find the right trade-off. We just used this pre-plot and then took the number of components that gave us over 90% of the variance. That gave us 5 components. And then if we look at the stretch behavior, we are comparing now the true curve. True is actually what we get from the simulator again by fitting the parameters to the data using then all the computational optimization in the space of the original model. So that is what is computationally expensive. And the blue line is what we get with the emulator. The shaded area is actually the 95% credible interval, because we can run MCMC simulations easily in the emulated space because it doesn't cost us anything. You see that the agreement is reasonable. Reasonable, by that I mean it is within the indicated posterior uncertainty, but obviously the agreement is not as good anymore as before because we have this higher dimensional space where we have to build the emulator. Here's another example of comparison between the simulator and the emulator. So the obvious question we asked was, can we do it better than PCA? But PCA is restricted to a linear manifold in this high-dimensional space. Can we sort of allow for nonlinearities? We tried all sorts of approaches, variational autoencoders, bottleneck neural networks, well, we didn't actually find any improvement. And I think the reason for that is that our training set is pretty sparse, only 150 examples. And so we needed to have a substantial amount of regularization in order to prevent overfitting, but that regularization pushes you more to the linear range of the transfer function. That didn't give us that much to exploit the nonlinear flexibility of the more nonlinear, more powerful machine learning methods. But then the question is, can we reduce the uncertainty by bringing in more prior knowledge? And that's what the next part is about. How can we include prior knowledge? We came across a paper where the physiologists had done the following study. They pumped blood into the left ventricle and looked at how the volume now inflates, how the volume increases as a consequence of different blood pressures. And that is shown here. So the pressure is at the vertical axis, the volume is at the bottom. And then they look at different patients. So the different symbols represent different patients. It's strange that they did it this way because really they changed the pressure. The pressure is the independent variable, the volume is the dependent variable. I would have done it the other way around. It's not surprising that the graphs are different, but then they came across the following fact that if they normalize the volume, they take the volume at a given pressure, divide by pressure at 30 millimeter mercury, and that is the volume at the unloaded state when you have no blood pressure, then they find that these graphs are all the same. And so that was the population of measurements they had got, and then they found this empirical law, an exponential graph. And so that's sort of an empirical finding. Can we use that empirical finding in order to improve our inference, improve our emulator? And so first we switched the axis because I didn't really like sort of the fact that the dependent variable was on the horizontal axis. So just going from this representation to that turns the exponential function into a log function. We also wanted to quantify the uncertainty properly to do Bayesian inference. So we realized that there's heteroscedasticity here, that the variance depends on the pressure. So we model that with the variance with a low order polynomial of the pressure. And so that is what it looks like as a probabilistic graphical model. We have now the Klotz curve behavior, that is the empirical finding in the paper. Just converting it from an x to a log by changing the axis. We have dependence on two parameters, alpha and beta, and then we've got the noise here. Okay, now if you put a volume at a given pressure, insert that into your model and solve for V30, then you can predict the volume at 30 millimeter mercury. And that's what we do here. We take the volume at 8 millimeter mercury, that is the blood pressure of a healthy volunteer. And then we can predict now the volume at the target level, 30 millimeter mercury. We can then use our original cardiomechanic model, which is trained on the volume of the left ventricle, as well as the 24 circumferential strains. And then it can also be used, once it has been trained, once we know the parameters, it can be used to predict the volume at 30 millimeter mercury. So we've got two different estimates, two different predictions of the volume at 30 millimeter mercury. They should be the same. We want to penalize any discrepancy between that. And there are different ways of penalizing discrepancies between distributions. We use a product of expert approach here. Kullback-Leibler divergence would possibly be the more natural measure, but the advantage of product of experts is that it gives us a closed form solution of this mismatch function. It's a function of the parameters. H is the geometry of the left ventricle. And then, well, we can't normalize it analytically, but we can run Markov chain Monte Carlo simulations to then get the posterior distribution of the parameters. If you look at the function here in the numerator, that's what it looks like. And the thing is that one of the parameters, theta2, is easy to infer. Theta3 is a parameter that is more difficult to infer. We have a higher degree of intrinsic identifiability as a consequence of the fact that we're only looking at circumferential strains, which don't give us a whole picture of the cardiomechanics. And the nice thing is that now with this prior, we can use inference of the parameter theta2 to reduce the uncertainty of the parameter theta3. That is basically what we found in our study. And when we're running simulation studies, we will look at the posterior distribution of the discrepancy of the inferred parameters from the ground truth parameters for this more difficult parameter theta3 for 6 different geometries. We see that we have a substantial reduction in the uncertainty compared to a uniform prior, and we're also getting closer to 0. Remember that 0 would be perfect prediction. We're looking at the squared discrepancy, the squared distance between the inferred parameters and the true parameters. Okay. So final question, can we go then beyond 25 dimensions to look at the full displacement field? And I think I can only give you a brief sketch here. That is where we want to use graph neural networks now. That is joint work with David Dalton and Hao Gao. So we want again to go from early diastole to late diastole. And we describe now the left ventricle as a graph where V is the set of nodes, epsilon is the set of edges. We have dependence on the material parameter that we're interested in. We also have dependence on the shape of the left ventricle. And we want to go from the initial state to the end state. So we can run a simulator, but the question is, can we do better By better, I mean at lower computational cost by running an emulator. So we are mapping the initial state into a high-dimensional embedding state. Then we do message parsing in this high-dimensional embedding state. Based on message parsing, we update the states of the nodes, and then we map that back into the original physical space. And that is our graph neural network. So we also use sort of shortcut connections to let messages pass through the system at a faster rate. It's being like on the motorway, you have a slow lane and a fast lane. That helps us with the message passing. So our nodes are located in the original physical space. The edges are in the physical space. The edges are just differences of the coordinates of the nodes that we connect. And then our state vector associated with the node is a vector that gives us information about the fiber orientation and an indicator whether it's on the boundary or inside. Then we map that into this high-dimensional embedding space. That has to be learned with the neural network, multi-layer perceptron. Then in the embedding space, we do message parsing. So we have messages. The message from node I to node j takes information from the state vectors of the nodes and the edge vector. Then we repeat that k times. It's an iteration. We use physical knowledge about the third Newtonian law of mechanics, that if two bodies exert forces on each other, these forces have to be of the same magnitude and point into opposite directions. So that's why you have this minus sign here. And then we update the edges by just adding the messages. And we add the node states by taking all the messages that feed into a given target node. And again, that gives us two functions that have to be learned with multi-layer perceptrons. Then finally, we do the decoder state. We map the embedding space back into the original state space to get then the shape of the left ventricle. And so again, that means we have to learn those neural networks. So we have overall 2k plus d plus 3 multi-layer perceptrons to learn. But d is the dimension of our displacement field. And so we then train the model by running forward simulations covering our parameter configuration space with space-filling design. We have several thousand forward simulations for different parameter vectors and different low-dimensional representations of our geometry of the left ventricle using a PCA dimension reduction. And then we train the neural network to predict then the states using the output from the simulator. Okay, so I think I'm running out of time now. Let me just show you sort of the upshot. What it gives us basically, it is a, we look at various other applications as well, but for the neural network, for the left ventricle, we're bringing down the computational complexity from, well, an hour with Phoenix. Okay, Abacus is not quite expensive, it's about 50 minutes, but we're bringing that down now to a sort of fraction of a second. So substantial improvement in terms of the computational complexity and at a reasonable displacement error. So the displacement error and then the actual displacement fields. Here we are comparing the result from the simulator with the result of the emulator. We haven't used that for inference yet. At the moment, we're only at the stage of building the emulator. We would hope that then the inference is going to be better because we, rather than just having 24 circumferential strain locations and volumes or low-dimensional 25-dimensional parameter vector, you now have this high-dimensional displacement field. But that's basically the next step. Following, building upon the emulator, our next step is inference, but we haven't got that yet. Okay, so let me just mention that this is work that has been done in SoftMaxSet. It's a research center funded by EPSRC. We've got 3 different centers, and this particular one focuses on emulation, uncertainty quantification, and hopefully eventually on translation as well.
Thank you, Dirk. I think we're going to get kicked out any minute now, so if we have time for 1 or 2 questions quickly. So I had a quick question just about the initial model of the ventricle, and basically how do you obtain that initial first 3-dimensional model before extrapolating it to the 17,000?
I mean, how we get the shape of the left ventricle.
Yeah.
Yeah, at the moment, it's the state of the art is the manual extraction. So you basically look at it, you draw various points at it. So a manual operator draws various points. Then we have a low cubic spline interpolation to then smooth the boundaries. We're currently also working on doing that with machine learning methods, automatic extraction, but the performance is not quite there yet. I think in general it's fair to say that if you have an experienced operator, that is the state of the art. But sort of the research done not only in our school but in other schools as well to do that automatically. So it's sort of catching up, but state of the art is still manual extraction.
Cool. Excellent. Thank you so much. Does anyone have any other questions?
So just about the 30 volume, the 30 mercury.
Yes.
What's so special about that value that makes it different?
That is what came from this study, this physiological study, that they were looking at sort of these experiments where they're pumping blood into the left ventricle, and they had just taken that as sort of as a reference point. We just want to see what happens if our model, after we've trained it, can we extrapolate to the same value, and then can we compare that with what came out of this empirical study, which was an empirical law that they found.
So you could use any value?
Well, yeah, I mean, it's just the empirical law was based on 30 millimeter mercury and the ground state. It was they're taking that as a reference point, so that was the empirical law. And for that reason, because we wanted to now use the empirical study from this physiological prior knowledge base, we wanted to also to extrapolate to 30 millimeter mercury. And there's a blood pressure you would have it in a living patient, or because that is sort of far beyond what you typically have in sort of a living patient in the order of 8 millimeter mercury. However, if you've inferred the parameters correctly, then the extrapolation can help you to resolve uncertainties about certain parameters. So it allows you to narrow down the uncertainties by looking at what would happen if you were to extrapolate that to higher pressures, which you can perfectly do in the lab. It doesn't happen in the human body.
Thank you. So I think we're going to get kicked out at any moment now. So yeah, thanks again to our two speakers, Colin and Dirk, and thank you again to all of you for coming along. So always have a nice rest of your day, even though it's pretty wet outside by the looks of it, and a nice long weekend. So thank you.